Um, okay, can you see my PowerPoint?
Yes, I can.
Okay, cool. Okay, so yeah, my topic is about gene therapy. Um, okay, so just like an overview of what I'm going to be talking about. First, I'm going to go over like mutations in the DNA, and then I'll go over what gene therapy is and its implications, so how it's being used, and then a couple of techniques and how the new genetic material is transferred to the patient, and then some of the risks and disadvantages that come with gene therapy, and the CRISPR-Cas9 system, which is a gene editing, it's a way of gene editing, and a success and failure story, and some ethical issues that arise with gene therapy. Okay, so for mutations, you are the way you are because of your DNA, right? But as amazing as it is, it does make some mistakes, and those mistakes are called mutations. And some of those mutations are good, while some are bad, and some show up while some don't. And so basically, when the genes are altered, the encoded proteins will not be able to carry out their normal functions, and that can result in genetic disorders. So there's two types of mutations. There's hereditary mutations and acquired mutations. So hereditary mutations are also called germline mutations, and that means that they're inherited from the parent and throughout a person's life in pretty much every single cell in their body. And and those ones are present in the germ cells, right? So the sex cells. And because they are present in the germ cells, they are passed on to future generations. There's also acquired mutations, so somatic mutations, and basically those are only in certain cells and acquired sometime during a person's life. And this could be through like environmental factors such as radiation or errors in DNA replication. And these somatic cells aren't passed down to the next generation. So that means that those mutations are only in that individual themselves. So future generations aren't affected by it. And some examples of mutations are cystic fibrosis, sickle cell anemia, Tay-Sachs disease, and color blindness. Like over here, if you look at the image here, there's three types of mutations. So there's substitution, insertion, and deletion. So in substitution, you can see that cytosine is being substituted with thymine. And because of that, an entirely new protein is going to be produced. Instead of the one corresponding to CAG, the one corresponding to TAG will be produced. And that will not result very well. And there's also insertion, right? So nucleotides can just be randomly inserted accidentally, and that won't result very well either. And there's also deletion. And in deletion, that's kind of self-explanatory, right? So some of the nucleotides are deleted and not all the needed proteins may form. And also, gene therapy does hold promise, kind of, to cure cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and things like that. Oh, one other thing I forgot to mention was that cystic fibrosis is actually a type of deletion mutation. So some of the nucleotides are deleted, and that results in cystic fibrosis. Okay, so what is gene therapy? So gene therapy is essentially the introduction of genetic material into cells. And basically, it's an experimental technique for treating disease by altering and correcting a patient's genetic material. And this is when the DNA is introduced into a patient to treat a genetic disease, and that new DNA that's being introduced usually contains a functioning gene to correct the effects of the disease-causing mutation. And there are two types of gene therapy. So there's somatic cell gene therapy and germline gene therapy. So that's kind of self-explanatory as well, because in somatic cell gene therapy, the foreign gene transfer is targeted towards a somatic cell, and then same for the germ cells, right? So in the germline gene therapy, it's targeted towards the germ cells. So really the main difference between those two is that germline gene therapy would affect future generations because it's in the germ cells, which are the sex cells, and then they'll be passed down to future generations, any changes made. And then also, gene therapy is really risky because, like I said, it was experimental, and it's also very expensive as well. Currently, the only way for you to receive gene therapy is to participate in a clinical trial. And clinical trials are research studies that help doctors determine whether whether a gene therapy approach is safe for people, and also helps them understand the effects of gene therapy on the body. Some uses of gene therapy are that they can replace missing or defective genes, and they can also deliver genes that speed the destruction of cancer cells, or even supply genes that cause cancer cells to revert back to normal cells, or deliver genes that stimulate the healing of damaged tissue. But yeah, that's basically kind of like the goals for that. Okay, so there are three techniques for gene therapy, and one of these is gene augmentation therapy. And this is the most common form, and it's used to treat diseases caused by a mutation that stops a gene from producing a functioning product or protein. And it does this by adding DNA containing a functional version of the lost gene back into the cell. And so that new gene produces a functioning product at sufficient levels to replace the protein that was originally missing. Um, and this is good for treating loss of function disorders such as cystic fibrosis because it'll introduce a functional copy that will then allow the cell to start functioning properly. So that's kind of shown in the image here, right? So you have a cell with a non functioning gene, and then you introduce that functioning gene, and then it starts working properly. Okay, so there's also gene inhibition therapy, and this is helpful for infectious diseases or cancer or inherited diseases caused by inappropriate gene activity. And basically, the new gene with the new gene will have a product that inhibits the expression of another gene or interferes with the activity of the product of another gene. And this is used to eliminate the activity of a gene that encourages the growth of disease related cells. So, especially like cancer, right? So, cancer is an overactivation of the oncogene, which stimulates cell growth. And so, this can be used to prevent further cell growth. So, in the image, you have a cell containing a faulty gene. And then, by introducing a blocking gene, you can block the faulty gene, and then the cell would function normally. Okay, and there's also killing of specific cells. So here, basically you would insert DNA into a deceased cell that causes the cell to die. And really there's two methods of causing the cell to die. So there is introducing a suicide gene that produces a toxic product which will kill the cell. So the suicide gene produces a toxic product, and then the diseased cell will die, really. And then you could also cause the expression of a protein that marks the cell so that the diseased cells are attacked by the body's natural immune system. So over here, right, so you have a diseased cell, and then you have your marker gene, and then the marker gene will cause the marker proteins to be expressed on the cell's surface, and then the immune system will recognize that and then attack the cell and then cause it to die. But what's really important in this is that you have to make sure that you're avoiding the death of the normally functioning cells. Otherwise, that's not going to end up really well. And this is also useful for cancer and trying to prevent that from spreading even more. Okay, and so basically, there's two ways of transferring genes to the body, right? So there's ex vivo gene therapy and in vivo gene therapy. So in vivo gene therapy, that it's basically the transfer of genes directly to the cells inside the body. And this is done when the individual cells can't be cultured outside of the body, or if reimplantation won't be efficient. And then there's also ex vivo gene therapy, where the transfer of genes, so it's basically the transfer of genes into the patient's cell outside of the body. And so the genes are transferred to cells grown in culture or in a controlled setting. And then those transformed cells are selected and multiplied and then reintroduced back into the patient. So if you look at the image, right, so you have for in vivo gene therapy, you have a vector. So the genetic material is carried through a vector, which I'll go into later. And then it's just inserted directly into the human. But in ex vivo gene therapy, you remove the cells and then you add in the genetic material through the vector and then insert it back into the patient. So, though ex vivo is more invasive and complex, and vectors aren't introduced directly into the human, safety check is possible, right? And close control is possible because it's in a controlled setting. Okay, and basically, vectors are carriers to transfer the desired gene into a target cell because to do that, you need a carrier. You can't just take a gene and just put it in the human and just expect it to work really. So you need a vector because that is the way of transferring the gene. And basically, a vector is a section of, so basically, a section of DNA or gene containing instruction for making a protein is packaged within the vector. So it's used for transport. And there are two main classes of vectors. So there's viral vectors and non-viral vectors. So some examples of viral vectors are the adenovirus, retrovirus, adeno-associated virus, lentivirus, and there's a bunch more. And then there's non-viral vectors such as electroporation, sonoporation, liposuction, and stuff like that. And there's like two types of non-viral vectors as well. There's physical ones and then there's chemical ones as well. Okay, so I'm going to go into two examples of vectors, so the retrovirus and the adenovirus. So normally viruses just bind to their hosts and introduce genetic material which spread as a part of the cell's replication cycle. So they hack the normal production machinery for the needs of the virus. But yeah, this is kind of like that, but not really. So for the retrovirus, at least, the genetic material is in the form of RNA molecules. But remember, the host cell, or because for humans, it's DNA, right? So the host cell is made of DNA molecules. So there needs to be a transfer from RNA to DNA. So when the vector infects the cell, it'll introduce its RNA together with some enzymes, and those enzymes are reverse transcriptase and integrase into the cell. And then it'll produce a DNA copy from its RNA molecule through reverse transcription. And so basically turning RNA to DNA. And if you remember, transcription normally is just DNA to RNA, right? Because transcription is part of protein production, right? Because you have transcription and translation. So you're turning DNA and you're copying that to an mRNA, right? But this is the opposite of that. So it's reverse transcription because you're turning RNA to DNA. And this is done by the reverse transcriptase enzyme. And then the DNA copy is incorporated into the genome of the host cell by the integrase enzyme. So yeah, it's kind of shown in the image here, right? So you have the viral RNA genome, and then through reverse transcription, you convert it to a double-stranded DNA, and then there's the formation of the pre-integration complex, and then it just enters the nucleus, and then the protein is eventually expressed. So this is really mostly targeted towards dividing cells because it is directly um, integrating into the genome. And so through cell division, right, it's easy for reaching multiple cells quickly. So that's a really good advantage, right? Because it's easy for the cells to contain new genes because of cell division. But a disadvantage of this is that if it is inserted into a wrong spot, like in the middle of another gene, it could cause cancer, right? Because it could get in the way of that gene and what it's trying to express, and it could really mess things up. Okay, so the adenovirus is another type of vector. So here, the genetic material is in the form of DNA. So it's not like the last one where it was an RNA and you had to convert it to DNA. It's already in the form of DNA. Except the difference is that it's not incorporated into the host's genetic material. So it's actually just left free in the nucleus of the host cell, and the instructions are transcribed like any other gene. But basically, the extra genes aren't replicated in cell division, and so it would need re-administration. So this is mostly targeting non-dividing cells because it's not being incorporated into the genome. Yeah, so if you look at the picture here, so you have the new gene, and then it's inserted, right? So it enters the cell through the membrane, and then After that, it goes through the nuclear pores and into the nucleus. So then the virus disassembles and delivers the DNA into the nucleus, where then the genetic information from the new gene is transferred to mRNA, which is the blueprint for protein production. So, one really good advantage of the adenovirus is that you don't need to be worried about inserting the new DNA into the wrong spot because it's not being incorporated into the host's DNA. Okay, and now for the non-viral vectors. So zone operation is a physical method, and basically this one, here you basically make pores in the cell with sound. So the ultrasonic frequencies deliver DNA to the cells, and so the with acoustic cavitation, it would disrupt the cell's membrane and allow the DNA to move into the cells, as it's shown here, right? So the plasmids will move into the cell. Okay, and then there's also liposuction, which is a chemical method. So the last one was the physical, and this one's chemical. So here, it's mainly based on the use of liposomes, which are artificial and have the same composition as a cell membrane with the hydrophilic head and hydrophobic tail. And they're used to deliver molecules like DNA into the cells. So over here you can see that the plasmid DNA is entering the cell, and then there's the lysosomal degradation. But really, one sec, I can't. Okay, yeah. So then the DNA is released into the cell, and then transcription occurs, and then you with the mRNA, right? So that's the new DNA is transcribed to the mRNA, and then translation occurs where the protein is produced. Okay, so some risks and disadvantages of gene therapy. So the immune response, right, is one major risk. So your immune system fights off intruders, but the new genes might be considered intruders because, and this can spark an immune response in the patient that can be harmful to them. So, with the use of vectors, that kind of reduces the likelihood of triggering an immune response, which is really important to avoid. And another risk of gene therapy is delivering the gene to the right place. So, the new gene has to reach the right cell. Otherwise, it could cause health problems. And even if it does reach the right cell, it needs to be turned on once the right cell is targeted because sometimes the cells can prevent the whole thing by shutting down genes that cause unusual activity. And this is a new gene that's being incorporated, right? So there is that possibility of that happening. And another problem is not disrupting, making sure that you're not disrupting the function of other genes. So, ideally, the new genes should integrate themselves into the genome and continue working for the rest of the person's life. But if it's inserted into the path of another gene, it'll disrupt its activity and could result in damaging effects in cell division regulation, which could result in cancer. And also, the high cost is a major disadvantage of gene therapy because it's very expensive. Also, that it's experimental, so you never really know what can happen. And additionally, multiple gene disorders can't be treated effectively with gene therapy. Okay, so the CRISPR-Cas9 system. So CRISPR stands for clustered regularly interspaced short palindromic repeat, and it's a gene editing technology used to modify, delete, or correct precise regions of DNA. And the CRISPR-Cas9 system is made out of the Cas9 enzyme and the guide RNA. So basically, what it does is it'll cut the DNA and then let the natural DNA repair processes take over. And this is actually really nice and helpful because it's faster, cheaper, more accurate, and more efficient than other existing genome editing methods. Also, the problem with this though is that the same ethical concerns arise with this as they do with gene therapy. So, like again, like targeting if you're targeting the germ cells, that could affect future generations, which is a major topic of concern. Okay, so something you want to avoid when you're using the CRISPR-Cas9 system is disruption, right? So if a single cut is made, a process called non-homologous end joining can result in the addition or deletion of base pairs, and that would disrupt the original DNA sequence and cause gene inactivation, which is something you want to avoid. But the CRISPR-Cas9 system is good in that it can delete unwanted genes or correct or insert new genes. So for deletion, right, a larger fragment of DNA can be deleted by using two guide RNAs that target separate sites. And then after cleavage at each site, the non-homologous ends can join, uniting the separate ends and then deleting the intervening or unwanted sequence. And then for correction or insertion, a DNA template can be added alongside the CRISPR-Cas9 machinery, and then that'll allow the cell to correct the gene or even insert a new gene using a process called homology-directed repair. Okay, so a success story is actually the first approved gene therapy, and this was done in 1990. So on a girl named Ashanti De Silva, who was 4 years old. So she was born with SCID, which is severe combined immunodeficiency disease. And basically, that meant that she lacked a healthy immune system, which meant that she was easily prone to infection. And so, what the doctors did was that they removed the white blood cells from her body and let the cells grow in lab and inserted the missing genes into the cells and infuse the genetically modified blood cells back into her bloodstream. And this was really good because it strengthened her immune system by 40%. So, if you can remember, right, so this is an ex vivo gene therapy method, right, because her cells were being cultured outside of her body, and then the new genes were being integrated within that, and then they were inserted back into her. So she's actually doing pretty good now. So that's why it's a success story. And the reason for selecting this disease for the first approved human clinical gene therapy trial was that this disease was caused by a defect in a single gene, and that increases the likelihood that the gene therapy will actually succeed because it's a lot less complex. Like if you remember when I was talking about the risks and disadvantages, right? So multiple gene disorders can't be treated effectively, and that's because they're a lot more complex than single gene disorders. Okay, and a failure story, unfortunately, is about Jesse Gelsinger, and he had a rare liver disorder and participated in the 1999 gene therapy trial. He died of complication from an inflammatory response after receiving a dose of the experimental adenovirus vector. So remember that one was the one where uh, the new genes were inserted through, it was actually DNA, right? So you didn't need, it was, I can go back to that slide actually. So the adenovirus vector is this one, right? So you have the adenovirus and it'll enter the cell membrane and then go through the nuclear pores, and then the virus disassembles and delivers the DNA to the nucleus. So after receiving that, he died of a complication from an inflammatory response. But his death actually halted all other gene therapy trials in the US for a while, and it sparked a much needed discussion on the regulation of experimental trials and reporting health problems. Okay, so lastly is ethical concerns. This is actually, I think, my favorite part of gene therapy because there's like there's so much question about this, right? Because each person has their own perspective on what they think about gene therapy and how it should be used and stuff. So there are a couple questions related to this. So the first one is, how can good and bad uses of gene therapy be distinguished? So that's kind of like basically who's going to draw the line between good and bad and whether it should be used for this purpose or not, right? And if it gets into the hands of the wrong person, right, that would not result well. So just kind of determining, the uses of gene therapy. And that also kind of relates to the second one, which is who decides which traits are normal and which constitute a disability or disorder. So, like whether gene therapy should be used for changing like personal appearance, or whether it's actually supposed to be used for something that prevents someone from doing daily tasks, um, and stuff like that. Also, will the high costs of gene therapy make it available only to the wealthy? Right. So, as I was mentioning earlier, gene therapy is not only very experimental, but it's very costly. So, if it does get available to the public, like, will only people who are able to afford it be able to get gene therapy? Also, could the widespread use of gene therapy make society less accepting of people who are different? Yeah, so unfortunately, we live in a society where not everyone is considered equal, right? So would would gene therapy kind of widen that gap, and kind of like would it so would people who get gene therapy, would they consider those who don't as less accepting, right? Are they not as good as the people who get gene therapy and things like that? And also, like, if people are getting gene therapy, and this also relates to like societal problems, right? Because if people are getting gene therapy to correct like things that they don't like about themselves, does that mean that like eventually people, it's I feel like it's going to end up in like a very like robotic society, right? Because everyone's going to be like the same, changing parts of themselves that they don't like, to make themselves more accepting in society, more accepted in society. Also, should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability, right? So if people could easily change parts of themselves that they didn't like, um, wouldn't everyone just basically be the same, right? Because there would be no uniqueness, right? Because everyone's going to be the same. Everyone's going to have the same abilities. Everyone might end up looking the same. And are we distorting nature, right? So, like, we were created so that we're all unique, right? And that's what our DNA does. Like that's what makes us all unique. And by altering that, like, are we distorting nature? Because since we're all supposed to be unique, we're all going to end up being the same if we change parts of ourselves that we don't like, which is a really important topic. And also, finally, germline gene therapy, right? Because so somatic gene therapy will only affect the individual themselves, while germline gene therapy will affect future generations. And this is a really big topic because people in the future generations, right, they have no say on whether they would actually want that gene to be changed or whatever, right? So it's like one person or whoever is receiving that germline gene therapy is making that decision for the future generations, which is the topic that kind of arises with like ethical concerns because they don't really get a say in that. But yeah, that's pretty much my presentation. If anyone wants to add like their opinions on any of these questions, feel free to do that. But yeah, that's it.
We are like almost at the cusp of like making major innovation in this field, and all those things are just really important to consider now, because the things our generation is pretty much going to grapple in almost for sure. So yeah, thanks again. Out of curiosity, I know Pia asked this, but do you guys have any questions for her before we wrap things up? All right, that sounds good. Thanks again, Pia. And I just want to make sure. So Daniel, you can end the recording now. I think